Hello, everyone. Good evening. I'm really sorry for keeping you all waiting. I was kind of stuck in traffic, so kindly forgive me for that.
You are forgiven.
Thank you.
You are forgiven. You don't have problem.
Hey, I can see.
All right. So I just want to find out one thing. With respect to your mid-semester exams, how was it like? Any feedback, any issues or whatsoever?
We manage.
Manage them now.
All right, cool. Okay, I could see patients.
Sir, please.
Our network wasn't responding, so we couldn't finish. Then it's submitted.
I understand. So that's why I understand. That's why I told you guys that got to get a good Wi-Fi in that regard when starting, because it's just a one-time thing, and when some of these issues comes, it becomes quite difficult for me to also handle. If I decide to give another attempt, then it means that I'm more or less like also being unfair onto those who attempted it once and all that. So whatever being the case, those who couldn't attempt it well, when I get the grade report, I'll know the way forward and know how to approach and all that. But for now,
Okay,
it's not against again. That's one thing. Okay, Regina, I could see that your hand is up. Kindly unmute your mic.
Hello,
Regina.
sir.
Yes,
Sir,
please.
good evening. Hello, sir.
Please go ahead. I can hear you.
Sir, please. So, as for me, I did about 3 or 4 or 5 and it submitted. So I called you, but you couldn't pick, and I emailed you.
So that's what I'm saying. At the moment, when some of these issues comes, it will be hard on my part to reopen. So I would personally get a grade report and know the mass apportion and know the number of questions each person attempted. If I realize there are some issues, then I'll know the way forward, how I'll be apportioning marks to some of these people who had such cases. The same thing happened to last semester. So, but if I am reopening, if there's no way I would reopen the exams again, or else we'll create some sort of unfairness and everything in here. All right. So I could see another person, Kasim Asana. Please, your hand is up. Kindly go ahead.
Please, sir, good evening.
Good evening.
Please, sir, my child was not well, so the time beat me. By the time I realized it was almost over, so I couldn't do. My child was admitted at the hospital, so I was just confused. I didn't know the time even overtake me, so I couldn't attempt anything.
I'm not exams now. Yeah, giving this excuse is not.
No, this one is not about excuses, sister. You are also a fellow woman. Please understand me.
So sorry about that, but you can see that you can ask. I know you've attempted other semester exams and all that, and if you will be really fair and honest enough, you can see that I give you ample time in that regard. I knew that some of these challenges might come up, so that was why it was an intentional thing that I did. I made sure that I reopened it on Thursday, and if I remember, it was there after either Saturday or Sunday, which was ample time.
Yes, sir.
Yes,
For that, you did
Saturday.
so
It was
well.
ample time, and I know some of my colleagues, when I some of my lecturers who gave only
We
2 hours.
appreciate it. You did
Please
so
Sir,
well.
keep quiet.
sir,
Allow
sir.
him to talk.
Oh,
Ah,
hello,
I know some of
hello.
my lecturers
Uh,
who gave you 2 hours for you to do the exams within that sort of time period. But I felt that due to network issues, if all of you I give you that same period, the system might jam. So that's why I graciously give you about 3 days or so, which I felt it was enough, ample time. And I gave the same period I gave to you, I gave to all other students, the regular students and other students that I'm teaching. So I felt with this, it was something I was not expecting too many issues or challenges when it comes to these things. So that's why I said there's no way I reopen whatever um issues that you come up with. Trust me, there's no way I will reopen the system again. But the key thing is that I will get a great report and know some of the challenges some of you had and know what to do in that regard. All right. So in order not to spend too much time on the semester, I'll give the first two students whose hand are up, then I'll continue on with what I'm supposed to do today. So I could see Kasim Asana. Your hand is up, so kindly unmute your mic and go ahead.
hello, sir. Sorry. Hello.
Please go ahead.
Sorry, sir. I
All
did.
right.
I'm sorry.
All right, no problem.
Yeah, I did. I'm sorry. I wrote the listen. I'm sorry. It was a different thing altogether. Sorry,
All
sir.
right, no problem. No problem. All right, so let me go ahead with what today I'm supposed to teach in that regard. So I think so. Let me ask the class where last time I got to, since I am. Oh. So please last time where did I got to? Please I think we've done this dendrite, soma, and what have you, if I remember.
Yes, sir.
yes.
Okay.
We
So
ended at the microglia.
glia cells. Okay.
Yes.
So we ended at microglia.
Yes.
Okay. Thanks so much. All right. So with regards to last week or last weekend, or let me say last week, what we did. Any questions pertaining to last week, what we did up to microglia? Any questions?
So if you can just wrap up, because some of us our network took us out the whole of the period.
Sorry about that. That wouldn't also work in that regard.
I did not even know where you started from, because I was actually going to suggest the oral.
That's
But we
one thing. It's a no-go area. So all right. So let me continue with Schwann cells. With Schwann cells, it has to do with the myelinated axons. Okay. So the last two glia, let me mute all of you and prevent others from unmuting themselves. Okay. Yeah. So with regards to Schwann cells and oligodendrocytes, these are the two main glia cells left. With regards to these two main types, when you check the nervous system, they are of the same structures, just that when that same structure is found in different areas, we have different names. It could either be Schwann cell or oligodendrocytes. Oligodendrocytes is normally referred to as oligodendroglia. So these two structures, Schwann cells and oligodendrocytes or dendroglia, they are of the same structure, just that when they are found in different areas, we have different names for it. And what structure am I referring to? They are the myelin sheath last time I told you about. So if you remember the myelin sheath that we went through. So let me get annotate. So there is a myelin sheath. If you remember, I told you when it comes to any neuron, there's a normal neuron without any myelin sheath. But some neurons, they have fatty and protein substances covering it. So there's the fatty substance covering this neuron. And that fatty substance is what is referred to as a myelin sheath. So when you see that, okay, a neuron like this has a fatty substance covering it, then it means that we normally refer to that myelin sheath as what? As either oligodendrocytes or oligodendroglia. Sorry, oligodendroglia or dendrocyte or Schwann cell. But for us to term it as Schwann cell, then it means that this myelin sheath needs to be, or this neuron needs to be at the peripheral nervous system. Right now, you know the difference between peripheral nervous system and the central nervous system. So that means when the myelin sheath of a neuron is found within the peripheral nervous system, we call that glia cell Schwann cell. But rather, when you find this same neuron with a myelin sheath covering it, and it is found at the central nervous system, which is either the brain or the spinal cord, then this time around, we wouldn't call it a Schwann cell, but rather oligodendrocytes, oligodendrocytes or dendroglia, which means the same thing. So these are the two main things. As I said, they are all the same structure. But when a Schwann cell, we call one a Schwann cell when it is found at the peripheral. And as I said, with the peripheral nervous system, any structure which is outside the brain and the spinal cord is referred to as the peripheral nervous system. So within the muscles, also, for instance, we have certain Schwann cells. Sorry, we have certain Schwann cells. And the Schwann cells are more or less like we have certain myelin sheath. So we have a neuron covered with fatty substances around the axon, which is the myelin sheath. And that neuron covered with myelin sheath, we call that myelin sheath what? A Schwann cell. But when you find the same myelin sheath within the brain or the spinal cord, this time around, we wouldn't call it a Schwann cell, but rather oligodendrocytes or oligodendroglia. All right. So these are the two main things you need to additional things when it comes to the the glia cells. So what are the functions of these cells, oligodendrocytes or Schwann cells? What are the main functions? Like any myelin sheath, we normally say that it insulates the cells. It insulates the cells in there. So generally, these are the main function. We have several functions of it, but know that, okay, any Schwann cell or oligodendrocyte insulate what they cell in that regard, generally. All right. So moving on. The next thing that we'll be handling is nerve impulse. So I'm moving ahead as I've always been doing. If you have any questions, you can use the emoji to raise up your hand. When intermittently, I will give you opportunity to ask your questions or seek for clarifications in whatever thing we are doing. All right. So the next thing has to do with nerve impulse. Right now, it means I'm done with the various parts of the neuron and the cells. And I'm moving back to a neuron. A neuron works in such a way that it transmits nerve impulses. It transmits nerve impulses. So we want to know within a neuron how these impulses works. When I say transmits nerve impulses, take it like, okay, it carries information. So a neuron carries information from one neuron to the other neuron. And the nerve impulse we are referring to, they are more or less like it carries some electrical charges in there. It has some electrical charges which transmit such information until it gets to the base of the neuron. Then it will release a chemical. So for it to transmit information, you want to know what are the main things within the neuron that helps us to transmit such information from one neuron to the other. So within a neuron, we want to find out what are the key things that goes on within the neuron that helps us to transmit such information. That's what the nerve impulse is all about. So certain researchers, scientists did certain tests just to help us understand how a neuron works and all that. And what they did was that they cut the axon. And currently, you know the various parts of a neuron, and you know that there's the axon. So they took a live neuron, cut the axon, like they divided it into 2 equal halves. Then they tested within the axon, what are the electrical charges within that sort of axon. Then also, when you check the outer layer of that axon, what are the key electrical charges in there? So per their studies, they were able to identify that within a neuron, after they cut that neuron, they realized that within a neuron, when the neuron, there's no nerve impulse. When I say no nerve impulse, when the neuron is not carrying any form of information, so it's like the cell is at rest. The neuron is at rest. Nothing is moving inside or inward. When we check the electrical charges within that sort of neuron, it has 2 main components. It has a lot of potassium in there. And also, it has a lot of other proteins. So other proteins means that the neuron inside has several proteins combined together. So normally in books, what I've indicated inside cell, some books will say the intracellular. So these are the terms sometimes they would use. Intracellular cell, which simply means inside cell. So it means that the intracellular cell has a lot of potassium, which is K plus, and other proteins, which is A minus. It has a lot of potassium and other proteins. So K plus means that the cell, it has positive what? Potassium, because potassium is positive. Its anion is positive. All right. Then when you check outside the cell, which is the extracellular cell, it has these 2 main charges. It has sodium. It has a lot of sodium in there, just positive, sodium plus, Na plus, sorry. Then it has a lot of what? Chlorine, which is negatively charged. So it means when the cell is at rest, nothing is moving inside the cell. These are the various electrical charges or ionic distribution. Within the cell, which is the intracellular cell, we have a lot of potassium and other proteins. Then outside the cell, we have a lot of what? A lot of sodium and chlorine. So usually, when you check the outer layer of the cell, which is the extracellular cell, when you taste it, it is salty. So that's what we are saying. It has a lot of sodium and chlorine. So usually, those who, I think you've done chemistry, or I think currently you're even doing biochem. So you know what I'm saying. When you combine sodium with chlorine, it means that's what you get. What do you get? Sodium chloride. And sodium chloride, it means that it is salty. So that's what we are saying. When you check the outer layer, when you taste it, it is salty of that cell. All right. So generally, this is how the ionic distribution is like when the cell is at rest, no movement, no information is being carried. So we normally say that when the cell is at rest, it is at rest. The cell is negatively charged. What this means is that inside the cell has a lot of other proteins. As I said, the inside cell has two main components: potassium, which is positive, then A minus, which is other proteins. So we normally say that when the cell is at rest, it is negatively charged to the point that it has about minus 70 millivolts. So it means that the A minus is too much to the extent that it has about minus 70 millivolts when you use a tool to check it. All right. Okay, so before moving on to the next gear, I think another one person's hand is up. So Ulera Damani. So please go ahead with your questions or clarification. Please unmute your mic. Please, you haven't unmuted your mic, so kindly unmute it and speak, or else then I would assume that is by mistake, and go ahead. All right, so I'm assuming maybe it's a mistake, so I'll go ahead in that regard. All right, so in most situations, these cells or these ions, sodium, chlorine, and what have you, each ion, that's sodium, chlorine, potassium, and other proteins, each of these ones have their own gate. They have their own gate that allows only that ion to pass through. So that's why I've used this to spell out how the gate is like. So when you check the green one, that's for chlorine. Sorry, that's for sodium. Then when you check the red one, that's for potassium. So this red gate allows only potassium to pass through. So let's say chlorine is coming. Chlorine wouldn't get opportunity to move into potassium gates. Chlorine needs to pass through its unique gate. The same thing applies to all the other ones. So we normally say that the cell is usually selectively permeable. So when you say something is selective, it means that it specifically will target only one ion and allow only that sort of ion to pass through. The rest, it wouldn't pass through. So that's why I'm saying each ion has its own gate that allows only that sort of ion to pass through. So that's what we mean by it is selectively permeable. Okay, others are annotating. So let me disable that. All right. Okay. So you remember I said usually the cell is at rest. And when the cell is at rest, we talk about how the ionic distribution is like. As I said, when it is at rest, the outer layer has a lot of sodium and chlorine, whereas the inner layer has a lot of potassium and other proteins. So the moment the cell moves from a resting state to a place where there's an action happening, there's movement happening, it means that something is really happening. It means that the ionic distribution also changes. So let me use a diagram to illustrate what I'm really talking about. I think this diagram pictures everything that I'm saying. So let's say this is this is more or less like an axon that we've cut. And what we are saying is that when we check the inner layer of this axon, for instance, the inner layer has a lot of potassium. That's why they've indicated KK here. Apart from that, it also has a lot of A minus, a lot of A minus here. So when the cell is at rest, this is how it looks like. Then the outer layer has a lot of sodium. So this is sodium, Na plus. Outside the axon or cell, it has a lot of sodium. Apart from sodium, it also has a lot of chlorine, Cl minus. So you find a lot of these things in here, Cl minus around this area. And all these things happens when the cell is at rest. When the cell is at rest, this is how it looks like. Then we are seeing that the cell wouldn't stay at rest forever. Usually, even you watching what I'm doing, there's a lot of neurons within your eyes and brains active doing these things that I'm saying. So it means that for you to even see, it means these neurons are being active, and when they are active, it means they've moved from a resting potential stage and they are doing something. So for it to move from a resting potential stage to a stage whereby they are in action, carrying information, these ions found at the outer layer and also the inner layer, there need to be some changes. And for some changes to happen, it has to do with two main forces. These are the two main forces in here. First of all, there should be concentration gradients. Then later there will be electrical gradients. So the concentration gradient has to do with something known as